The overall goal of this method is to determine the HDAC inhibitory potential of compounds and to identify selectivity towards HDAC6 or HDAC1 in cells using a single ultra-high performance liquid chromatography mass spectrometry assay.
This method can help answer key questions in the drug discovery field, such as determining whether an HDAC inhibitor is selective for HDAC6 over class 1 HDACs in living cells. The main advantage of this technique is that it requires a single incubation step with cells, inhibitors, and selective substrates in order to determine potency against two HDAC isoforms simultaneously.
The implication of this technique extends toward early drug discovery of selective HDAC inhibitors because it detects whether HDAC6 selectivity is maintained in living cells, avoiding endogenous protein acetylation assays.
So, this method assesses selectivity of HDAC inhibitors in HeLa cells. It can also be applied to other cell lines such as primary and other tumor cells, including clinical specimens.
Culture HeLa cells to 80% confluency in a T75 cell culture flask in minimal essential media supplemented with 10% fetal bovine serum, penicillin G, and streptomycin. Wash the cells twice with 5 milliliters of Dulbecco's phosphate buffered saline. Then aspirate the buffer and add 1 milliliter of cell dissociation reagent. Incubate at 37 degrees Celsius for 5 minutes to detach the cells.